Hey everybody, we're here at the Plant Science Research Center on the campus of Auburn University. It's one of many centers conducting research on peanuts. We'll be learning more about that and how we're helping produce a better peanut for the industry. Hey everybody, we're here at Auburn University's E.V. Smith Research Station in Tallahassee, Alabama, and we're here with Dr. Sands. So Dr. Sands, tell us a little bit about what you do and what we're going to see today.
Thank you very much. So I'm Dr. Alvaro Sanz. I am a crop physiologist. I am now an associate professor, and in this field where we are testing is how peanuts respond to drought and how we can make a peanut more drought tolerant.
Okay. Well, can you show us a little bit about the work that you're doing with peanuts?
Yes, let's go and I can show you.
Okay, cool.
So in this location, in this experiment, we are running a drought experiment where we are testing how different cultivars of peanut respond to drought. We have done many years of research, and Dr. Chen started with this many years ago, where he tested about 200 lines of peanuts, and he started to select the peanut cultivars for drought tolerance just by testing which cultivars had higher yield under drought. Now, what we are doing as a physiologist, we want to understand why these cultivars have higher drought tolerance, because if we can understand why they have higher drought tolerance, we can try to breed for it, because if we understand the characteristics that makes that cultivar drought tolerant, we can transmit it and pass it from one cultivar to another and have cultivars that they are more drought tolerant. Also, what we are trying to understand is what are the mechanisms of drought tolerance, because there is no only one mechanism for drought tolerance. There is many ways for peanut cultivars to be drought tolerant. For example, having deeper roots, having stomatas in the leaves. The leaves, they have little pores that open and close. There is some cultivars that we are testing here that they are very good at closing very quickly the stomata, so they transpire less. So we want to see if there is different mechanisms of drought tolerance, and understanding how they work, later we want to try to see if we can combine those mechanisms together. Because if we can stack up several mechanisms of drought tolerance together, we can make like a super peanut that is drought tolerant.
Peanut,
yeah,
right?
the perfect drought tolerant peanut. Or if we cannot do that, because it's not very clear that that is possible, we can try to understand what drought tolerant characteristics make that peanut drought tolerant in different conditions. So here under the rainhouse shelters, this structure, this kind of greenhouse, what we are able to do is to simulate drought. So whenever it rains, all the plastic close, and we are growing the plants under drought, and we are taking different measurements that I want to show you today. One of the measurements that we are doing is taking photosynthetic measurements during several days and testing the maximum capacity of photosynthesis of plants, and that's what Sajid here, my PhD student, is doing.
Okay.
And maybe we can go and walk and talk with him, and he can show us what he's doing.
So introduce yourself and then tell us and show us what you're doing here.
My name is Sajid Hanif. I am a PhD student from Pakistan. Today we are using a liker, and it's used to measure the photosynthesis of the plant. And today specifically we are measuring the plant response to different levels of CO2, like how different cultivars they respond to different levels of, like we have minimum CO2 level starting from 50 ppm to the 1500, to see like how different cultivars respond to elevated CO2 do they respond differently or at different levels, or they behave at the same level. So we are checking to that one.
And what is measuring is the amount of CO2 that the leaf is getting and how much water is getting out. So it measures how much photosynthesis can do, how much CO2 can get, and also how much water is transpiring. Because plants, when they do the photosynthesis, they need to open these little stomata that they have in the leaves, these little pores. So when they open them to get the CO2 in, they have the problem that they are using water, they are losing water. And so they have the dilemma that for growing they need to get the CO2 in to do photosynthesis, but at the same time they lose water. So that is what we are testing a lot in this experiment. There is cultivars that they are much more efficient and getting more CO2 with less water, and that's what we are testing, and that is what this machine does. The other mechanism is how plants get water, and usually plants and peanuts get water through the roots. That's why we have these tubes in here. These are access tubes for a machine that is called a mini rhizotron. So we have these tubes going 4 feet into the ground, and we are able to open this tube and put a root scanner that goes all the way at different depths and takes pictures of the whole tube. And with that we are hoping to see if there is differences between cultivars in how they develop the roots under well water and under drought conditions.
Yeah. So can you show us this field and how it might differ from the drought?
Yeah, so in this field, this is our well water control treatment. So we call it well water not because only is outside and it doesn't get covered by rain. We call it also the well water because you can see that it has irrigation lines. So while the others, they get covered and they don't get any water, these guys are water every week to basically supplement all their needs. So these peanuts, you can see that they are taller, they are greener, and if you check in the soil, they are much more covered and they have a lot of more leaves. You can almost not see any soil available. In the others, you might see a little bit. Other thing that I want you to notice when we compare with the drought ones, in here all the leaves are pointing to the sun. You don't see any leaves that they are flipping or trying to basically run out of the sun. In here, all the leaves are upright, pointing the sun that now is just on top of them. So they are happy, they are doing maximum photosynthesis and growing as much as they can. When we go to the other field, to the drought field, to inside of the rainout shelters, we will see how the symptoms of drought stress. So here we can see the differences between the well water plants that they were outside, that they were a little bit taller, but the main difference when drought starts to set is how they start to roll the leaves. So you can see in here how the plant is rolling the leaf and basically is getting wilty, so is losing water in the leaf, so that makes the leaf to kind of roll. It's doing it because the plant is losing water, but also because it's getting so stressed right now that it does not want to receive a lot of light because it cannot do photosynthesis. So that extra light that cannot be used for photosynthesis can damage the leaf. So that's why plants under drought, like these ones in here, they start to roll the leaves, and you see kind of these wilty symptoms, and that's indicative that the plants are getting dry. You can see this usually at midday and in the late afternoon. We are going to see it now, starting now is when probably every day they are going to start showing more and more these symptoms. We are also taking notice of this with drone imageries. We are taking pictures every day at midday, so we will be able to see at different days how the different cultivars are showing these symptoms or not. And cultivars that they last longer in showing these symptoms, they are probably more drought tolerant than others that not. So that's why we are taking these observations.
So at night, once it's turned its leaf, will it turn it back over at night?
Yeah, at night usually soil water comes a little bit more up and there is no such high temperature and that much light, so the plant recovers a little bit of water. So at night and in early morning, the majority of them, they look fine until they get super stressed. If we come in 2-3 weeks that we are going to maintain the experiment, probably after 3 weeks more of drought, they are going to be all wilty and much worse than this, even in the mornings. But it's a process that it takes days and days to do. So in the second part of this project, Dr. Chen is trying to understand what are the genetic mechanisms of this drought tolerance. So we have identified cultivars that they use more water because we think that they have deeper roots. We have identified also drought tolerant cultivars that they close very quick the stomata and they save water. So we call them water savers and water spenders. So he is doing genetic and genomic experiments to try to understand what are the genetic mechanisms of this.
So you're taking both of those to like gene whatever and like just molding them together to make this super duper peanut that can adapt both ways, like the water.
We want to try to understand if that is possible.
That's possible.
Dr. Chen thinks that is possible. I think that is not.
Hey everybody, we're here at the Plant Science Research Center greenhouses and we're here with Dr. Charles Chen. So Dr. Chen, tell us a little bit about who you are and what we're doing here today.
Okay, yeah, I'm the Dr. Charles Chen. I'm the professor at the Department of Crop, Soil and Environmental Science, but my specialty is working on the peanut breeding. In terms of the breeding program, we established that in the Auburn University since 2011. The major purpose is to develop a new variety can adapt to Alabama State, also for the southeast area like Florida, also Georgia State. And the main goal is to genetically improve the peanut yield for farmer, also like a disease resistant, and also has the drought tolerance character, so adapted to our Alabama condition.
Okay, well, can you show us inside the greenhouse and what you're doing?
Yeah, sure. Please come in. Yeah.
So Dr. Chen, can you show us a little bit more about your process with breeding?
Okay, yeah. Like I say, the breeding program usually has 3 phases to develop a new variety. Phase 1, we are doing the crossing. So that means you will find 2 different genotype, or we can say they carry the different trait. Then we want to integration those desirable traits in the one variety. And so the first phase is doing the crossing. You can see the greenhouse here. In this greenhouse, we have 70 pots growing here. So we have probably about 20 different genotypes. So that means that 20 genotypes, they carry in the different desirable trait. For example, someone has the high yield, but not good disease resistant. Another line has the good disease resistant, but not a high yield. How you going to integrate those 2 desirable traits together? Then we'll go through the processing, doing the crossing. That is what we are doing here. We grow in those different genotype in here. Until they get in blooming, so that is the time doing the crossing. Because the peanut flower is a complete flower, That means the one flower here, they have the male part, also the female part. So before the flower blooming, they doing a self-pollination. By nature, but as a breeder, we needed to go to do the pollination based on our design. Then we can harvest that hybrid and take into the field growing the next season. And after that, plant growing up, we call the F2 seeds. So the F2 population, all the trait, they go into segregation. So some plant carrying the high yield, some plant carrying the disease resistant. By the way, as a breeder, we were looking for either the both have a high yield and disease resistant. And from there, we were going to call the phase 2 is breeding selection. In my program, our breeding headquarters is in the Heartland Station. So we have 1,000 breeding lines growing there for selection. So that will take probably about 5 to 6 years until generation 6. So genetically, each plant becoming homogeneous. So they no more segregation. So, and then from there, then we go into the third phase. It's called the preliminary yield trial. Because we selected that plant only in the one location. We don't really know when you are growing in a different environment, how the plant performance over there. So then we go into a couple of years working on the yield trial. That means just to see our breeding selection has a good performance in different environment. So for example, in the south of the Alabama state, or in some of the like middle area. So anyway, it's try to check what is the G by E in the reaction. G by E means genotype by environment in the reaction. So after that performance test, then we get a better idea which line going to release. And then we release for the farmer. That's the whole process. It takes about 10 years to 12 years to finish the breeding program.
Yeah, to get your end product and the goal
Yes.
that you maybe set out for.
Yeah.
That's a long investment.
Yes, yeah.
Yeah. Well, can you show us how you actually cross it? Like how you actually do it?
Actually classes.
Yeah,
Okay. So like I say, we go to do the emasculation. You need to find the very tiny young flower like that. It's still not blooming. If you see the flower like that already blooming, they self-pollinated. It's failed. It's too late. So the first is identify that flower, and usually I use a magnify, easy to manipulate it. So you can see here, first one, you need to open the flower. It's a tedious work, or you need to very gentle. Don't hurt the stigma. So you remove that, and then you will be going open the flower. You can see after open, you can see here the essence. You have a 9, that is the essence. We will remove that, you see?
Mm-hmm.
So that's about 9 total. So you see, after remove all the pollen, the essence, the very, like I see here, that is the stigma. So these are, we already done emasculation. What are we doing now is I need to collect the pollen. So for example, I will use this one as a male. So we just collect the pollen. So after you find the male parent, open this one. So on the leather tips, they have a pollen. You can see the pollen. You can see that pollen, and then we take that one, coming back, putting the pollen on the stigma, the tips there. You see? So you put in there. After that one, how we going to label that? So you will use the different color of the strands. So like my toolbox here, we have different color that's designated to different parents. For example, we got a flower from here. This one is number 2 parent. So then you can need to label the flower. So then you can get the strands. Then we label that one. So this the flower we already pollinated. So we just waiting for 5 to 7 days. And that if they successful fertilizer, then they will going to grow in the peaks. So you can see, I can show you here, that the pollen and fertilizer, the over will develop in peaks. And then peaks will penetrate to the soil. So after like 5 to 7 days, I need to coming back. If that flower succeeds, then I need to put the label. So I put in the label on that packs showing 10 times 5. That means the parent 10 closest with parent 5. So then after they are mature, so I will harvest that hybrid. So I take that hybrid next season, grow in the field.
Okay, got it.
So this kind of job is very tedious job, but it's very critical to succeed of the breeding program.
Once the students leave your lab, how are they going into the peanut industry, and maybe what are they doing? Do you know?
That's a good question. Like I say, mainly, like for the undergraduate student, a lot of them are going to pursue the advanced degree, and like working on the master's degree or PhD degree. And if for the other student, they were looking for the job in the USDA, because the advantage for our breeding program is we have a very tight collaboration with the USDA ARS National Peanut Lab. So oftentimes I also bring that student working in the peanut lab. So somehow they know something what's going on the USDA the research lab. So they will give the student more opportunity. They know maybe they are understanding that part. They will be triggered the desire to working on the USDA. The student, they are going to do the industry.
Hey everybody, we're here in Headland, Alabama at the Alabama Crop Improvement Association, and we're here with Brandon Dillard. So Brandon, tell us a little bit about where we are and who you are.
Yes, hey, I'm Brandon Dillard, Auburn grad in 2005 with my BS, and worked here in Headland at the Alabama Crop Improvement Association, and started here about 5 years ago. And so we're housed here in Headland, but we cover basically all of Alabama. And basically what we do is we take breeder seed from the plant breeders, and we increase it so that it's available for farmers to buy.
Okay. So, for example, you would have Dr. Chin's seed, and he
Yes.
would bring a little bit of it to you, and then
Yes.
you would grow it out, right?
Yes, yes, absolutely. So Dr. Chin, some of his peanut varieties that we do here, he'll bring us 50 pounds, 100 pounds, 500 pounds, whatever. And our job is to take that and plant it in the fields and increase it, and then shell it and have seed available for more increase and for research and for the farmers.
Okay. So can you show us a little bit about the process? Walk us through the process when he brings you the seed. What's
yes,
the next
absolutely.
step?
Yeah,
All
let's
right.
go.
Okay. So what is the first step?
So the first step is obviously in the field. We grow those peanuts out. So here at Alabama Crop Improvement, we are contracting with farmers, and then we oversee that growing process, and we have them do the growing of it, the spraying and things like that. And so they'll bring those peanuts into us out of the field. We go out and clean their harvest equipment so that there's no seed in there, because our main goal when we're increasing seed is to make sure that that variety is 100% pure. So when Dr. Chin gives us a variety, that may be the only 50 pounds in existence. And so our job is not to screw that up. And so we try to keep that pure, and that starts in the field by us overseeing, making sure that the planters are clean when they're done. So once all that's done and they're harvested and those peanuts roll into here, our first step is to get them off of a semi onto these peanut wagons and the drying process. Now, when it's seed, the drying process is very important. You can over dry them, you can under dry them. And so our job is to make sure that we don't get them too hot and that we get them at the perfect moisture to store into the warehouse.
And that just keeps them from getting molded?
That's correct,
okay.
yes. Mm-hmm.
All right.
Absolutely. And then we can go look at the farmer stock warehouse if you'd like and see
Okay.
where we store.
Yeah.
Okay, so this is the peanut farmer stock warehouse. So as we get those peanuts in from the fields, we'll store them in this warehouse. Now you can see this is not like a regular warehouse because it's just a really small compartment, but we have 8 different compartments. And like I've said, our goal is to keep them pure, and so we have to make sure that we don't cross-contaminate any of the varieties. And so on the end of the warehouse is a pit that we dump the peanuts in, and it goes up top into that belt. And so we set what we call the kicker, and those peanuts fall into this. So whatever variety we're dumping, we move that kicker to the bay it's in. And then, so we'll fill, by November, this thing will be all, you could probably step off that up there at the top onto the
And the
peanuts.
peanuts?
Mm-hmm.
Oh.
All right, so after we bale the peanuts out, our next step is to shell those peanuts and get them ready to treat and bag for seed. And this is our shelling operation here. A lot of lingo that can go through, but this is a 3-stage sheller. So that means we have 3 different sizes that we shell. And so when you harvest peanuts, you'll have anything from a big jumbo down to a small peanut. And so with our shelling operation, we need to shell the big ones first, then mediums, and then the small ones. So anyway, so we have a 3-stage sheller, and we shell those peanuts, and then we separate the rocks and sticks and stuff from them. And then we go to what we call an electric eye, which has a sensor in it, senses any kind of damage to the seed, or if the husk is gone off of it or something, the skin is off. It uses air, and it senses that bad seed, and it kicks that bad one out. And so that has saved a lot of time and labor. It does a really good job. And then we are unique that we have 2 belts inside that room that people sit on both sides of those belts, and they pick out anything that shouldn't be there or any seed that looks bad that the electric eye might have missed. And so commercially people don't do that. We do that just to be sure 100% that we're getting pure seed out, good seed. So, and then after that, we're going to bag them, put them in the warehouse, and then we'll pull germ samples and purity samples, and the high oleic ones we send off to get a high oleic oil sample done to make sure. That helps us to know that those are pure, and that also that we did a good job growing them, that the high oleic oil content is where it needs to be also.
So talk to us about the different levels of certification.
Absolutely. So basically we get the breeder seed, and that's your top class. You know, like I said earlier, it may be 5 seed, it may be 50 pounds, it may be a ton, but either way that's denoted as breeder seed. We take that breeder seed and we grow it, and the breeder may come and do some selections off of that to make sure it's what he wants. But our first stage is foundation seed, and that's what we sell. We use the breeder seed to create foundation seed, and then we put it out on the market. So your larger seed companies such as Sessions or Birdsong Peanut Company or Golden Peanut, they'll buy that seed, that foundation seed, and then they have 2 stages they can go down to. They'll grow that foundation seed out and sell registered seed. All right, and so then they can also take that registered seed, put it back into seed production, and produce certified seed. And so that's our last stage. Most of your growers are buying certified seed. It has been through a protocol to maintain a level of purity. And we also do that in our house for Florida and Alabama. Southern Seed Certification Association is housed here, and that's where our inspectors go out and look at those foundation seed, registered seed, certified seed to make sure they're going by the rules and that seed is going to be a pure seed when it's put in a bag after selling.
That's pretty cool. So it's maintaining its integrity through certification and then
absolutely, that's right.
ready to go.
Yep.
That's pretty cool.
Yep, absolutely. That's our job here. We grow those seed out, and then we inspect them to make sure everybody's going by the rules and doing a good job.
Yes. So what kind of opportunities for students, like job opportunities, are for them in the industry?
Yeah, so you know there's massive opportunity in the seed industry. I mean, anywhere from the breeding side all the way to the sales side. You know, that's one side we hadn't even talked about. Was a lot of times we sell to companies that sell that seed, and so they may grow some out and sell it to growers or whatever. So there's a lot of between making the machinery, building the warehouses, operating or working on equipment, to actually growing the seed, being an agronomist, to being a sheller man, to being an inspector in the fields. I mean, it's basically limitless of where you can be in this operation.